Hello friends, this video on cell cycle and cell division part 8 is brought to you by examfear.com. No more fear from exam. That is anaphase. Now in this phase, what happens is the sister chromatids become free. What do we mean by free? That means they are no more attached or they are no more linked to each other. They become free. So free chromatids are pulled by kinetochores towards opposite poles. So now you will get to know what happens. Have you ever played the game of tug of war? What happens? You have a rope, you have two teams on either side and both the teams pulls the rope towards itself. Correct? Now what do you think will happen if suppose the rope is not very strong? Suppose if both of them are exerting very high forces on both the ends, what might happen? The rope might break in the middle. That is quite possible, right? Now, a similar concept applies here as well. Here in this case, these spindle fibers are connected to the kinetochore on this side. Again, this spindle fiber is connected to the kinetochore. So, in both the ways, the spindle fibers start pulling the sister chromatid towards itself. As a result, the two sister chromatids which were connected so far get separated. So if you consider the picture, it was something like this. This was one sister chromatid, this was another sister chromatid, and this was the kinetochore. So this kinetochore was connected to this side, this was connected to this side. Now this exert pressure in this direction, this exert pressure in this direction. As a result, what happens? It breaks from here and one goes this side, the other one comes this side. That is what is happening. If you see, this is going this side, this is coming back this side. So this is how the sister chromatids get separated. Now once the sister chromatid gets separated, they are no more called as sister chromatids. They are known as daughter chromosomes. So they migrate towards opposite poles due to the interaction of the kinetochores and the spindle fibers. Now, who is responsible for this motion of the sister chromatids? This motion happens due to interaction of kinetochore with the spindle microtubule, because if the bonding between kinetochore, that is this one, and this spindle fiber is not strong enough, it will not be able to pull and separate the sister chromatid from the chromosome. So that bonding has to be really strong. So that is one thing that happens. So one thing is the sister chromatids get separated and they are pulled towards the opposite poles by the kinetochores. That is one part of anaphase. What else happens here? The cell elongates. How the cell elongates? Now while I was talking about the spindle fibers, I missed to tell you one thing. Now there are two types of spindle fibers that are formed. Now when the spindle formation takes place, it happens that some of the spindle fibers are not attached to any of the kinetochores. Let us suppose if this is how the spindle fiber is formed. Just for example, I am telling you. Now let us suppose there is a chromosome here, there is a chromosome here, there is a chromosome here. So if you look at this spindle fiber, it is not attached to any kinetochore. So this one is known as kinetochore spindle fiber and this one is known as non-kinetochore spindle fiber. So there will be a few fibers which will not be connected to any of the kinetochores. So what will happen to those fibers? Because of the presence of those fibers, now the kinetochore spindle fibers will separate the sister chromatids and try to pull them towards opposite poles. But the ones which are non-kinetochore spindle fibers, they are still there. So because of the presence of those non-kinetochore spindle fiber means they are microtubules. So those non-kinetochore microtubules will make the cell elongate. So these sister chromatids will move towards opposite poles. So these sister chromatids will exert pressure this side, this will exert pressure this side, but at the same time this non-kinetochore microtubule is also there. So somebody is pushing it this side, somebody is pushing it this side, and somebody straight is standing here. So what will happen as a result? So a force on this side, this will move slightly this side, a force on this side, it will move slightly this side. As a result, cell elongation will take place. So So I hope the concept is clear. There are two types of microtubules which form the spindle fibers. One which are connected to kinetochore, that is kinetochore microtubule. Because of the kinetochore microtubules, the sister chromatids get separated and they move towards opposite poles. The second type of microtubule is the non-kinetochore microtubule, which are not connected to any kinetochore. Because of their presence, the cell elongates during anaphase. So these are some of the things that take place in anaphase. Now from this you can get a very clear idea that what happened. Basically what you did in the beginning, that is during the S phase of interphase, the duplication of chromosome happened. So the chromosome which was like this happened to be like this. So now you have two copies of the same chromosome. Correct? Now let me give you an example when I say two copies of same chromosome. Let us suppose you get one set of chromosome from your mother which says that you should have black eyes. You get another set of chromosome from your father which says that you should have blue eyes. Now both of them are for eye color. So basically both these chromosomes are for eye color, but they are two copies of the same chromosome. Correct? So two copies. So one is say blue color and one is black color. So they are basically two different traits of the two parents. So but it is the same thing. It is the eye color. So now here also the similar thing happens. So the same trait gets distributed into two halves. Like how I gave you the example of making Xerox and then distributing the notes. That is what we did. We prepared the Xerox, or we did the photocopy during the S phase. And now in this Anaphase, what are we doing? We are distributing the photocopies. So this was the photocopy because both represented the same thing. It was just replicated. So now we are separating them. So we'll give this to one daughter cell. We'll give this to another daughter cell. So that is the concept of mitosis. Replicate and then divide. So now the last phase, that is telophase. What happens in this phase? Now if you see, you already got to know that okay, you have got two different sets of genes on two different ends of the same cell. So now all you need to do is you need to divide that cell. But there are few things which are missing now. Because the nuclear membrane was gone, the cytoskeleton was gone, but you need all those things in a cell. So what happens in telophase? Nuclear envelope redevelops. So here if you see, nuclear envelope redevelops around the genetic material. Genetic material was nothing but the sister chromatids or the daughter chromosomes. Now please remember that after anaphase, the sister chromatids, once they get pulled to opposite poles, then they are known as daughter chromosomes. So please do not get confused with these different terms. You should know that the sister chromatids, as long as they are together in a chromosome, they are called sister chromatids. But as soon as they get separated, they are called daughter chromosomes. So now, wherever you have your daughter chromosomes, here and here, a nuclear envelope will develop around that. Spindle will disassemble, so the spindle will vanish. Now the same microtubules will be used to make up the cytoskeleton. Chromosomes will disassemble, again the chromosome will also be gone. So what will come into picture? Again those chromatin threads will come into picture. Actin filaments form cleavage at the center of the cell. Now there are some protein filaments which will form a cleavage, a structure like this, a cleavage like this at the center of the cell. Now this cleavage will deepen further and this will actually help the cell to get divided completely. However, that will not happen in this stage. In telophase, what will happen? Nuclear envelope will come back, spindle will vanish and the cytoskeleton will come back. Again, the chromosomes will vanish, the chromatin material will come back. So the chromatin material will tend to get collected in mass in the two poles. So here you will have some chromatin material, again here you will have some chromatin material. So the chromosomes is lost now. Other cell organelles, for example, nucleolus, Golgi apparatus, endoplasmic reticulum, all these cell organelles will start to redevelop. So how this actin filament forms the cleavage, that we will see in the next phase, next stage, that is cytokinesis. So here all the different cell organelles will also develop. So now you see, you can actually see the two daughter cells getting formed. It is just that the cytoplasm is still connected. The nucleus have already divided. So the karyokinesis is over with telophase because your nucleus is completely divided. So you have two separate nuclear membrane, which clearly states that the nucleus have already got divided. And now we will see the last phase, that is the cytokine. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.